had some delays. I was going to say something else. Oh yeah, in lab, you'll have your first unit exam this week, and then next week you should start working on Unit 2 content. So I will be a little ahead, but here's what's going to happen, right? So I'm going to talk to you about lymphatics today. In lab next week, you will work with some models and identify some major lymphatic vessels, groups of lymph nodes, and things like that. And then I'll kind of start talking about immunity. Immunity is not covered in lab. Okay, so immunity you'll only hear here in lecture. So once you're done with lymphatics there, then you will actually get ahead in lab because you'll start the respiratory system, the anatomy portion there, okay? All right, so let's go ahead and kind of cover some of these concepts here. So for the lymphatic system is, okay, so it is also a network of vessels. These are not blood vessels, these are lymphatic vessels. And so they're organized. I mean, you got the same kind of designations as such. You don't have arteries and veins, they're all lymphatic vessels, but we will talk about capillaries and larger lymphatic vessels like collecting lymphatics. And then of course they all congregate, they kind of like meet at a set of lymph nodes. So we're going to talk about the role of those lymph nodes as well. Today's lecture is mostly slides. I won't use the board a whole lot. Next lecture it gets very animated, but for today it's mostly the slides. Okay. So we'll talk a little bit about this. When you think about this network of vessels, it is not a closed system like the circulatory system was, meaning, of course, you have a starting point, right? And your smallest lymphatic vessels are capillaries, very similar to your blood capillaries. But then as they become larger and larger vessels and they become networks of vessels, ultimately they all drain fluid within the lymphatic vessel, which is called lymph. They drain all that lymph into the venous circulation. So it is kind of, if you start at a point, you don't come back to the same point like you did with the cardiovascular system. So it's why you don't really say circulation. It is a lymphatic system, okay? Okay, so we'll talk about all of these different types of vessels, and you will hear about it and see examples of this in lab as well next week. So the main lymphoid organs, meaning organs that are related to the lymphatic system and therefore related to immunity, are mostly what you see here, and you will identify these in lab. The spleen, the thymus, tonsils, very important. Lymph nodes, different lymph nodes basically have different names based on how they are regionally located. And then, of course, we'll talk about some specialized lymphoid tissues like mucosa-associated lymphoid tissues, MALT. Okay. All right, so here are some of those lymphoid organs that you'll be identifying in lab. So the tonsils is a group of three different tonsils that we need to talk about. They are associated with the nasal cavity and even the oral cavity. So of course, it's a screening point for air that you're breathing in case there's any pathogens that need to be trapped and eliminated, right? And then of course, it's a screening point for food and drink that you're consuming through your mouth, okay? Then the thymus, this is a very important organ. This is where a population of lymphocytes called T lymphocytes, they undergo an education or a maturation process. So I'll definitely come back and talk about the thymus probably next week when we talk about T and B cells. Spleen, that's a very important organ that plays a huge role in detoxification, but also has an important role to play lots of different things. In relation to RBCs, this, along with the liver, will help with the proper breakdown of those RBCs, taking care of the hemoglobin and all of that. But there are other roles too. And then you have these congregates of lymphoid cells called Peyer's patches, and these are associated with the small intestine. You're going to see this mostly in the distal portion of the small intestine in a region called the ileum. And this is also another screening point for, again, any kind of bacteria or contaminants within the food as they pass through the entire digestive tract. At the junction of the small and the large intestine, you have this organ called the appendix. So this is, again, another screening point for bacteria or any other pathogens that need to be eliminated. So you will go into some details in lab. You will see cross sections, histology views of several of these organs, most certainly the thymus, the spleen, Peyer's patches for sure in lab as well. So you'll see what it looks like on the inside as well. All right, so when you think about the lymphatic system, all right, so this is a topic that's near and dear to my heart. This is This was my PhD dissertation related to the lymphatic system. I was looking more at a developmental aspect of these lymphatic vessels and how they guided function. One of the unifying concepts that we talked about when you took ANP1, very first chapter, is this idea of complementarity of structure and function. If you recall, the form or the structure of a particular organ is what's going to dictate how it performs functionally, right? I mean, what is its role? Its role is guided by how it is structured. And so it's actually, this is something that we saw development of these lymphatic vessels prenatally, right? I mean, as they developed because they had a functional role to play, right? And that's what differentiated them into like the very simplest capillaries versus the ones that are very important for lymph transport, which would be the larger lymphatic vessels called collecting vessels, where you have to have muscle cells there to do that job of lymph transport. So I'm going to keep it very short here. Like this is one of those topics that unfortunately I wish I could talk to you more about, but no, we're going to cover this very, very quickly because really what this kind of leads into immunity. And that's what I want you to understand is what are some of those key lymphoid organs? What are their roles? What do they look like? And then what that kind of leads us into the whole immune system. Mostly I want you to keep in mind what types of cells you're going to start seeing in these organs, right? Because these cells will have a very important immune role to play. Okay. All right. So some of the functions of the lymphatic system, really, it's the biggest functional role is that, yes, it's not very glamorous, but it is the sewer system of the body. Okay. Meaning any leaked fluid from your blood vessels as they accumulate within your tissue beds, right? I mean, that's going to cause edema. And of course, it's accumulating in your tissue beds, which means your blood volume might go down. And we know if blood volume goes down, then that can cause imbalances in blood pressure too, right? If blood volume goes down, blood pressure also goes down. So really what the lymphatic system is doing is making sure that that blood volume is fairly stable, right? And preventing edema. So any excess fluid that's leaked out of blood vessels and accumulating in your tissue beds, we want to return it safely back into the blood circulation. So that's why the most important role of the lymphatic system is returning interstitial fluid that's been leaked, right? But there's a lot of other immune cells that are constantly moving around in this lymphatic network of vessels. So therefore, very closely tied in or integrated with immunity as well. So when you're thinking about this network of vessels, the smallest vessels are called capillaries. So this is very similar to blood capillaries. Got to think back to what that looked like. So blood capillaries were your exchange vessels. This is where exchange of gases and nutrients occurred between the blood and the tissue beds, right? And you'll also cover this in lab when you did capillary filtration, right? Same idea. Except here, this is not important for exchange of gases or anything, but structurally very similar to those blood capillaries. So the wall of these capillaries will only have a single layer. Remember, you still have that tunica intima, which is that innermost layer, simple squamous epithelium, which is called endothelium. Same deal here, right? Single layer. This is then going to become larger vessels called collecting lymphatics. These are like your larger arteries and veins, right? But collecting lymphatics resemble veins more so than they do arteries. Okay, so again, lower pressure kind of vessels, very large, larger lumen, right? And yes, they also have valves. Okay, to a certain extent, especially in yeah, for the most part. Then if you're thinking about these collecting lymphatics, again, like arteries and veins, you have a in endothelium layer, then you have a muscle layer, right? All right, so blood vessels, arteries and veins, they have smooth muscle. Collecting lymphatics, it's muscle. It's not exactly smooth muscle. It's actually a hybrid, very, very unique, very interesting. They combine characteristics of cardiac and smooth muscle. So it's really called lymphatic muscle. It's its own breed, okay? Um, Functionally, same role, right? Contraction, relaxation, which helps with the movement of lymph. And again, when you're thinking about this, very similar to veins, we're having to return leaked interstitial fluid, right, everywhere in the body. Again, you're returning it back towards the heart. Okay, and I'll tell you, it's right, kind of right up here in the neck region. So you're having to do this against gravity. That's why valves, like the veins did, right? As these collecting lymphatics pump kind of blood, not blood, lymph, they unite to form larger trunks, and I'll show this to you. And then these trunks ultimately become one of two ducts. So you have your thoracic duct and your right lymphatic duct, and I'm going to show it to you on the next slide. What you see here is real, okay, on the top here, this is basically a connection, red, the arterial side, blue, the venous side. And then here at the tissue beds, this is where you're seeing any leaked fluid here, right, that needs to then be pulled into those lymphatic capillaries, from there into those larger vessels. You've got a whole bunch of different lymph nodes, right? And then returning it back into the venous circulation. Okay, so all the green portion there is basically your network of lymphatic vessels. Zoomed in there, that's actually a lymphatic capillary. I think the next slide goes into more detail. So let me go ahead and talk about that. Yeah. So the lymphatic capillaries, like I said, very much like blood capillaries, they're much more permeable though. Got a single layer of cells, the endothelial cells. They do have openings between those endothelial cells that almost look like little valves. I'll tell you in a second. And then those valves are anchored to the extracellular matrix. There's a lot of collagen filaments. These are called anchoring filaments that helps to kind of hold that lymphatic capillary together. Specialized lymphatic capillaries within the digestive tract. So this, you're going to see this in the intestine, right? The specialized lymphatic capillaries called lacteals exist. And so they play a very specific role. Okay. No, not doing that. Sorry. Okay. Lacteals are specialized lymphatic capillaries within the duodenum, specifically of the small intestine, and they're going to play a role with absorption of lipids. We're talking fatty acids and other nutrients after they've been digested down. So I'll come back and talk about this with the digestive system, which is not this unit, but the next unit. And then we're going to talk about how they form something called chyle. So keep that aside for right now, but that's a very specialized lymphatic capillary. All right, so this is what a lymphatic capillary looks like. This is a single layer of cells, the lymphatic endothelium. And so each of these cells have these gaps, or these valve-like kind of structures, which allows for fluid from this brown interstitial space basically to enter this lymphatic vessel. As soon as this fluid enters the lymphatic capillary, now it's called lymph, right? So really it's pressure differences here as well, and I'm not going to go into those specifics, but really it's very simple. It's when the pressure in the interstitial space, when it is greater than the lymphatic capillary, those valves, those mini valves kind of open, right, allowing fluid to enter this lymphatic vessel, forming lymph. And then as lymph starts to accumulate here, we're kind of closing those valves here, preventing backflow. And then of course, this is going to lead into larger vessels, and it's going to flow away from the tissue bed. These are those collagen fibers, those anchoring filaments that help to keep those lymphatic vessels anchored or tethered to the surrounding extracellular matrix. Okay. All right. So then, all right, as these lymphatic vessels become larger, these are called collecting lymphatics. So again, like I said, very similar to veins, right? Thinner walls, you still have the same three layers. You have the endothelium on the inside. You have muscle. This is lymphatic muscle in the middle layer. And then you've got collagen reinforced, like fibrous connective tissue on the outside, forming the externa or the adventitia. These collecting lymphatics then become larger trunks. A lot of these are paired trunks, meaning you'll have a left and a right, correct, for the different sides of the body. You have paired lumbar trunks on the bottom, and then you have your bronchiomediastinal in the kind of like the thoracic region, subclavian neck, jugular, subclavian, well, sorry, armpit kind of in that region, jugular neck. And then there's one single intestinal trunk related to all the intestinal regions, okay, or the abdominal regions. So I'm going to go ahead and show this to you. But again, in lab, you will identify these different trunks. Now, all of these trunks will then unite to form one of two lymphatic ducts. Your biggest lymphatic duct is called the thoracic duct, okay? Your right lymphatic duct is a little smaller, and it's where this drainage is coming from, okay? The right lymphatic duct is collecting leaked interstitial fluid, right? Only from the right side of the face, the neck, the right side of the thorax, and the right upper limb. Does that make sense? Just this right portion. lymphatic drainage from this right side. They all kind of drain through different trunks, ultimately into this right lymphatic duct, which means lymph from everywhere else in the body, right? The left side of the head, neck, thorax, and abdominal downwards, everything drains into that, the thoracic duct, right? So what are we doing? We're returning leaked fluid. We're returning excess fluid back to the blood ultimately. So therefore, both of these lymphatic ducts, they empty into the venous circulation. And notice this is at the junction of the internal jugular and the subclavian vein. So if you all remember your blood vessels, right? On the right side, we've got my subclavian vein and then the internal jugular, right there at the junction of that internal jugular and the subclavian vein. This is where I'm going to see the right lymphatic duct drain into, right? At the junction on the same side, left side though, right? You got the subclavian, left subclavian and the left internal jugular, correct? At that junction, that's where you have the thoracic duct leading into. Does that make sense, right? It's the same junction. It's the internal jugular, right? And the subclavian vein. Okay, so here's showing you the thoracic region. There's the trachea, the esophagus at the back, and you can kind of see your major veins there. Your superior vena cava, your inferior vena cava. So again, all of these vessels here in green are your lymphatic vessels. Those enlarged structures are nodes, right? So kind of showing you here, you've got your intestinal trunk, which is just a single trunk. And then these two trunks are your paired lumbar trunks, the left and the right. They unite to form. So you got the two lumbar trunks and the intestinal trunk that unite to form this enlarged region here called the cisterna chyli. And then from here, exiting the cisterna chyli, that green vessel is the thoracic duct, right? And let's trace it. Thoracic duct goes all the way through the abdominal thoracic region, and you see how it merges right there, correct? That entry point is what? The left subclavian vein, that blood vessel, and that's the left internal jugular vein. You see it, correct? So that's my entry point for the thoracic duct. And then the same thing's happening on the right side. So here's the right internal jugular vein and the right subclavian vein, correct? At that junction right here, this is where you're seeing the right lymphatic duct. Does that make sense? All right. Let's talk a little bit more about some of the, oh, sorry, I went too fast. Okay. How do you propel lymph? Lymph is the fluid that you're transporting through the entire network of lymphatic vessels. There's a lot of different things that help with lymph transport. Okay, remember it's very similar to veins. So it's a lower pressure kind of system, and you're transporting a lot of that fluid back to, again, in the neck region right here, correct? So which means you're having to do this against gravity. That's why you've got valves. But then you also have, well, first of all, you have, so it says smooth muscle cells in the walls of these lymphatic, collecting lymphatics. But remember, they're not exactly smooth muscle cells. It's really it's a combination of smooth and cardiac properties. So yes, contractions of those muscle cells helps with return of that lymph back to where it needs to go. But you have a lot of other extrinsic factors that also helps this. Again, very similar to veins. So as lymphatic vessels get squeezed, right, between skeletal muscles as they're contracting, that's going to help with return of lymph. Thoracic changes related to pressures, related to respiratory movements, that's going to help. The valves, we know that's going to prevent backflow as we're trying to return all of that lymph against gravity. And then, of course, arteries in the vicinity as well can help with return of that lymph, right? So it's got its own muscle cells that will help with the return of lymph, but you have a lot of extrinsic factors that are helping that movement as well, okay? All right, so now let's talk about these cells. Different types of lymphoid cells that you see within lymphatic structures or lymphatic vessels and then ultimately lymph nodes. So your major population of cells are your lymphocytes. And this is where we're going to spend a whole lot of time the next two lectures talking about B cells and T cells. So this is all related to what we call adaptive immunity. And they have very different responses. B cells, when they get activated, they are going to basically differentiate into a population of cells called plasma cells right here, which then release or secrete proteins called antibodies. And these antibodies can then go mark a foreign pathogen and basically mount an immune response against it. The thing about T cells and B cells is that it's very specific. Every B cell, every T cell on its surface, it has a receptor, right? It has a receptor that is programmed to only recognize one foreign antigen, one particular strain of a bacteria or virus or some kind of pathogen, right? It's recognizing one specific target, and when it recognizes it, it binds it, right, forming a complex. And then that's basically going to then mount an immune response against that foreign pathogen, right? B cells would work by releasing antibodies. These antibodies can then recognize that foreign pathogen anywhere in the body, right? So let's say, I don't know, let's say a B cell in a lymph node in my arm region, right? I mean, it recognized a foreign pathogen. It marks that foreign pathogen, right? Which means those antibodies then get released into the bloodstream. It gets released into the lymph, which means now we're going places. Now we're able to recognize that foreign pathogen anywhere in the body. So this is what I call a systemic response, right? So very specific, but systemic. Same thing with T cells. When the T cells get activated, it's very specific. It has a very specific receptor to target a foreign antigen, and then it mounts an immune response. T cells do not form antibodies, right? The way that they work is a little different, and we'll talk about all of this in the next two lectures. T cells could work by activating something called cytotoxic T cells, which can go target that foreign pathogen that it's recognizing and then destroy it, right? Or it might work, there might be a different type of T cell population called helper T cells, which does a whole lot of different things, right? And then, of course, you have your regulatory T cells. So T cells don't work by producing antibodies. That's B cells. But again, their roles are very specific, and how they actually function, each of those. B cells is an 80-minute lecture, and T cells is another 80-minute lecture. So you're going to have to wait for that, right? So we'll come back and talk about all those details in the next few lectures. So the main type of lymphoid cells you expect to see as they circulate in lymph, as they go through the collecting lymphatics, and as they screen lymph nodes, and as they go through all the different tissues, we're looking for, the T cells and the B cells are looking for foreign targets that they can basically mount an immune response against, right? Those are your specific cells, though, but there's a whole lot of other cells, right? There's a whole lot of other cells. And so you've got some that are a little bit not as specific. And you've heard of macrophages in ANP1, right? And remember, macrophages actually came from monocytes, and monocytes was a type of formed element, right? Okay. So when monocytes arrive at a site of injury or inflammation or whatever, the monocyte gets specialized, meaning it differentiates into these guys called macrophages. Macrophages are non-specific. All they're looking for is we're recognizing an antigen profile on the surface of every cell in the body. So every one of your cells, right? Epithelial cells, fibroblasts, muscle cell, kidney cell, liver cell, whatever. Every cell has a profile on its surface. Do y'all remember the plasma membrane? We talked about something called like a sugar coating on the plasma membrane called the glycocalyx. Okay, some very tied into that, right? Very similar to that. It's a recognition pattern that tells your immune cells like macrophages and B cells and T cells and other immune cells, this belongs to me, right? I mean, if it's a normal liver cell, if it's a normal muscle cell, the profile I'm going to express on my surface is going to be self antigens, meaning it belongs to me, it belongs to self, correct? So which means if a macrophage is patrolling the body and it encounters a liver cell with these self antigens, it should say that's self antigen. I recognize it, don't touch it. Don't mount an immune response because that cell does not need to be destroyed. Does that make sense? But then if you had a foreign pathogen, like a bacteria or something, right, a pathogen that's invaded your body, well, obviously the recognition profile, the signature pattern on its surface is not going to be self antigen, right? It's not something that your macrophages have ever recognized. It's something that's invaded your body. It's new, it's foreign, it's non-self. Does that make sense? Right? So then your macrophage goes, well, that is not a self antigen. That's foreign. That's non-self. I don't recognize it. I don't care what strain of bacteria or virus or whatever you are. You don't belong here. That's all I know, right? Which means then that macrophage, it's why it's non-specific. All it's saying is that foreign antigen? If it's foreign antigen, I'm going to mount an immune response against it. How do macrophages work? That's coming up in the next few slides. We've actually talked about it in ANP1, process called phagocytosis, right? Then you got these cells. We're going to talk a lot about those. Those are called dendritic cells. Dendritic cells are what we call antigen presenting cells. Their job is not to mount an immune response against, like, it's not to go after a target and destroy the target. Its job is to patrol the body, right? And its job is to identify foreign antigen. That doesn't belong in here, right? And then I'm going to place markers on it or basically sound the alarm saying, I found something here. It doesn't belong here. Now all of your specialized cells come do your job. Does that make sense? We're just sounding the alarm. So what it's doing is it's presenting a foreign antigen, right? That's why it's an antigen presenting cell. It says, look what I found. This is what that foreign antigen looks like. T cells, B cells, macrophages, all of y'all need to come in here, identify this foreign target wherever it is in the body and destroy it. Make sense? Okay. Then most of your lymphoid organs have T cells, B cells, macrophages, dendritic cells, all these other types of immune cells, right? But you got to have like a stroma, like a glue that holds everything together. This is your reticular cells. That's basically your foundation. And you'll actually saw this tissue in ANP1 lab, that very first unit when you'll discuss different tissues, connective tissues. Reticular connective tissue was a variety that you saw. And you most likely saw a cross-section or histology view of a lymph node, where you saw that reticular connective tissue, lots of fibers. You remember those reticular fibers, all of these fibers that were like very chaotic, very branched, right? So lots of fibers with these reticular cells that's forming the foundation, right? So then kind of trapped or residing within that network, within that stroma is the B cells, the T cells, the macrophages, right? Some of them are resident, but most of them are not just going to sit there, right? They're going to keep moving. That's their job, is immune surveillance throughout the body, okay? Does that make sense to y'all? All right, so that's your overall view of what a lymphoid tissue looks like. So let's go into some examples here. Any of your lymphoid tissues, again, this could be lymph nodes, great example, right? That's an example of a lymphoid tissue, spleen, thymus, any of these. You're going to see that a huge variety of lymphoid cells, right? A lot of these tissues are going to have lymphocytes in them. Now, there's some variations, I'll explain it to you, but most of these, when you think about your classic lymphoid tissue, like a lymph node, you're going to see macrophages and dendritic cells, and you're going to see B cells and T cells, like an entire, variety of those lymphoid tissues. So I'm going to break this down. Two main types of lymphoid tissue. You have some that are very, very specialized. Their role is immunity. Their role is lymphatic in nature, right? These are called lymphoid follicles. That's their job, right? I mean, that's their main purpose. Then you've got other tissues. We call these diffuse lymphoid tissue, where this might be a digestive organ. This might be a respiratory organ. This might have other roles, but you have a population of cells within that organ that play an immune role, that have an immune role. Does that make sense? So that population of cells is what I call diffuse lymphoid tissue. So I'm going to show you what a classic lymphoid follicle looks like, germinal centers, all that fun stuff on the next few slides. Let me explain it to you. I'll show it to you in relation to lymph nodes. So in lab, you will identify different populations of lymph nodes, like axillary lymph nodes and cervical lymph nodes and inguinal and mesenteric and so on and so forth, right? Really, it's a group of nodes, and they have a regional name, right? So you know all of your regional terms. Hopefully you remember it. So it's going to make sense as you start looking at this in lab. What are the main roles of these lymph nodes? So the main role is that, as you can see, you have different lymphatic vessels that lead into this population of lymph nodes. And then in the lymph node, we are actually filtering all this lymph that's coming through, right? We're filtering the lymph to see if there's any pathogens in it, and if there's anything that we recognize that we want to mount an immune response against, right? Anything that is foreign. So that's amazing. Most important role is filtering lymph, right? And then, of course, if you do identify a foreign pathogen, then you have to activate the immune system. And that's a very broad term, right? Activate the immune system would mean activating different arms of the immune system, innate immunity versus adaptive immunity, okay? So what does the lymph node look like? Here's, again, you will see this in lab next week. You will see a cross-section under the microscope of what that lymph node looks like. So I'm going to break this down for you. Two main regions. cortex versus the medulla. So a lot of this is going to resemble very similar to the adrenal gland. You'll remember that. The adrenal gland, which is on top of like the kidneys. You remember this, right? So it had an outer region called the cortex. And then as you go deeper and deeper, it becomes the adrenal medulla, right? Same setup. The lymph node has a cortex region on the outside, and then it becomes the medulla on the inside. Keep that in mind because it's the same anatomical structure for the kidney. And then a little later on, when we talk about the reproductive system, ovaries, yeah, it's the same idea of the cortex on the outside, the medulla on the inside. Except here, of course, the cells that you see here within those lymph nodes are not going to look like what you see in the kidney versus the ovary, right? So let's talk about that. I'm going to go ahead and show you my next slide and explain all of this right here. This is, I guess, a schematic of the lymph node in cross section. So you have an outer capsule region that's like the casing, right, that holds this entire thing together. This outer region here is the cortex. The deeper portion is the medulla. And you notice how the cortex has all of these compartments. So you've got these thread-like kind of structures called trabeculae that is kind of invading into the cortex, forming all of these compartments, right? All right, so these compartments, this is the cortex region. At the very center of the cortex, right? This entire region here is called the lymphoid follicle. And the center of the lymphoid follicle is a deeper staining region, and we call this basically the germinal center. So the cortex consists of different compartments, and within each compartment, you're seeing a group of these cells which stain together as like one unit. This is called a lymphoid follicle. And then within the lymphoid follicle, the center of it is called the germinal center. So what type of cells will you see here? You'll see a whole variety of lymphoid cells, but what those germinal centers are important for is B cells, okay? So you have your immature, rather, you have your unactivated B cells, right? I mean, they're programmed, they know what to do, but they haven't yet encountered a foreign pathogen, right? We call these naive B cells. So you have a lot of naive B cells that are constantly moving through this lymph node, go to the next lymph node, and so on and so forth throughout the body, correct? But what if a B cell here encounters its foreign pathogen, right? As that lymph is coming through, and we're identifying a foreign pathogen, and that B cell says, I have the receptor for it on my surface. I know I'm supposed to recognize that particular foreign pathogen. I know what to do with you, right? So then when that B cell gets activated, activation of those B cells is happening here in those germinal centers. Does that make sense to you? So typically when you think about the cortex, the germinal center, it's not the T cells, it's the B cells there, right? Then of course, as the activation process continues, you're going to see those B cells are going to migrate inwards into the medulla region, and then they have to go out those those lymphatic vessels. Does that make sense to you? But which then means you will also see T cells, macrophages, dendritic cells, and a lot of other immune cells here within the medullary region. Okay, let's talk about lymphatic vessels. On this side, this is the incoming side. You're seeing 1, 2, 3, 4, 5 different lymphatic collecting vessels, right? In comparison, in the hilum region, which is where the exit point is out of that lymph node, you have 2 outgoing vessels. So you have many more incoming lymphatic vessels than you have outgoing vessels. Do you see that? Correct? So this can cause a backup of lymph, right? And this is intentional. You have more incoming vessels than outgoing vessels because you want that lymph to kind of back up here in the lymph node, because that is important for recognition of your screening. You're looking for any foreign pathogen for the whole activation of those appropriate immune cells. That's why you want lymph to stall in that lymph node. So you have more entry points, fewer exit points, and that allows for filtering, efficient filtering of lymph within the lymph nodes, okay? And that's explained here. Why do you have, okay, efferent is outgoing, afferent is incoming. Okay, so why do you have more incoming vessels than you have outgoing vessels? All right. So far, so good? All right, so lymph node, that's a very important lymphoid structure. Let's talk about a few other structures. Spleen. All right. Spleen plays a huge role as well in basically proliferation of those lymphocytes. They play a role in overall activation of the immune system, so therefore surveillance. But it also has other roles. Spleen, along with the liver, plays a role in elimination of any kind of detox, right? So any kind of aged cells that need to be removed, but specifically RBCs, right? Along with the liver, you remember how the RBCs had hemoglobin in it? Hemoglobin consists of the amino acids, the globin chains, which is not a problem, but then you have the heme part with the iron. The iron can be toxic, right? So the spleen and the liver have efficient ways of storing back that iron, and then, of course, bringing it back into the circulation when needed, when it needs to be incorporated to form more hemoglobin, right? And then there's a few other roles for the spleen, but what we talked about is probably the most important ones. You probably will see a cross section of the spleen in lab. All right, so there's really two big regions here. You have a white pulp region and a red pulp region. White pulp is where you're going to see all of your lymphocyte cells. This is going to be important for activation of an immune response. But then you know how the spleen is also important for cleanup or degradation of the RBCs and safely storing away the iron and all of that part, right? So all of that is handled in the red pulp region. So red for red blood cells and what needs to happen there. White for the white blood cells and how you're activating the immune system there. So those are the two main regions. Okay. Thymus. All right. When it comes to the thymus, there's only one lymphocyte population here. It's nothing to do with the B cells. So thymus, this is kind of how I remember it. T for T cells. Okay. So this is a very important site where T cells become immunocompetent. So again, I'll spend about 20-25 minutes talking about immunocompetence next week and what that really means. And that's basically an education process that needs to happen for those T cells to become immunocompetent so they can recognize that foreign antigen and then mount an immune response against it. That is happening in the thymus, okay? So this is very, very active in your early years of childhood. After all of your T cells are programmed, your thymus does not really have a functional role, and it can become smaller and smaller and atrophy as you age, okay? Okay. Thymus plays a role in maturation of T cells, but also this other population of T cells, a specific population called regulatory T cells, very important for autoimmune disorders. So when regulatory T cells, this is very broadly speaking, when regulatory T cells start malfunctioning, then there's like so many subpopulations of these Tregs, right? But based on where the abnormality is, that we think is the premise behind, like, development of autoimmune dysfunction. Okay. All right. So then you'll definitely see a cross section of this in lab as well. I'll have Ms. Gosnell kind of walk you through that. And the last piece is this mucosa-associated lymphoid tissue. What does this mean? Throughout the body, you're going to see collections of these lymphoid cells or lymphoid tissue. Again, these are not specifically lymphoid organs, right? It's not like lymph nodes or the thymus, right? Does not have a specific lymphoid or lymphatic or immune kind of function. It is a different organ. It has a digestive function, or it has a respiratory kind of a function. But there are collections of these cells that form what we call a lymphoid tissue, right? What we call a mucosa-associated lymphoid tissue. What does that really mean? Okay. All of your internal organs that are hollow, right? So wherever you see like a lumen, right, where something is moving through it. So this could obviously be passage of air. It could be passage of food if you're talking about the digestive system, right? Think mostly about the digestive tract here because I'm going to explain a few. So thinking about how that food will pass through different digestive organs. As we get into the small intestine, so the mucosa is basically the innermost lining that's directly in contact with that lumen, right? So if you're looking at a digestive tract, as food is passing through and getting digested, different enzymes, the mucosa is the lining, is the innermost layer of the wall of the digestive tract that is directly in contact with the food, right? And normally this is some kind of epithelium. Okay, and of course, it's going to vary as we start talking about the respiratory system versus the digestive system, but it's some kind of epithelium, right? So associated with that lining, the mucosa, there are congregates, or basically there are patches of what I call mucosa-associated lymphoid tissue. MALT, right? So if you see malt tissue related to the small intestine, especially the distal portion of the small intestine, this is called Peyer's patches, right? We talked about this lymphoid related tissue at the junction of the small and the large intestine. That's the appendix. Then you have a group of malt tissue called tonsils. This is kind of shared by both the nasal and the oral cavity. So it has, like, based on its location, respiratory or digestive functions as well. Okay. So the main role of malt tissue, all right, what's the main role? The main role here is it's acting like a screening point. So if you think about the tonsils, as you breathe in air, right? So we're screening the air. If it's any bacteria, any pathogens, we're basically sounding the alarm, right? If you're thinking about the digestive tract, then of course it might be contaminated food or drink that you have consumed, right? Okay. So let's quickly go over this. The tonsils, there's 3 collections of tonsils, and I think you may, no, probably not. In lab, next unit, you'll start working on this. You have your pharyngeal tonsils. These are your adenoids. This is related to the nasal cavity. Then you have 2 groups of tonsils here that are more related to the oral cavity. So you've got the palatine and the lingual tonsils. So again, the pharyngeal tonsil is a screening point for air. As you breathe in air and it goes through all of these structures here called the nasal conchae, as you swirl that air around the nasal cavity, the pharyngeal tonsils is your first line of defense, which is going to sound the alarm if there's any bacteria or any pathogens that you breathed in, right? So then what does it do? The tonsils don't mount an immune response against it. It's just going to say, we've got something that needs to be taken care of. Then you've got your macrophages, your neutrophils, and other cells that will respond to this invading pathogen, right? Likewise, if you consumed any food or drink that's contaminated, and you have any pathogens that need to be eliminated, then you got your palatine and your lingual tonsils that would do the same job, right? All right, then a little further into the digestive tract, in the wall of the small intestine, specifically the distal small intestine, which is a region called the ileum, this is where you have patches of malt tissue. This is called the Peyer's patches. So again, this is my screening point for bacteria, right, or any other contaminants as that food is being digested and passes through the GI tract. At the junction of the small and the large intestine, you have the appendix, which kind of does the same, performs the same role. Okay, so I think we've covered everything related to the lymphatic system. Yeah, yes, ma'am. So no, different. So the Peyer's patches is associated with the innermost lining, the mucosa, right? The lacteals are specialized lymphatic capillaries that you see within the small intestine, within a structure called the villi. I'll tell you that. Their role is, the lacteals don't play a role really in immunity as such. They're part of the lymphatic lymphatic network. They're absorbing something, right? Typically, when you think about a lymphatic capillary, we're pulling in excess or leaked fluid, right? The lacteals have a role in absorbing digested lipids like fatty acids and monoglycerides and glycerol and things like that. So slightly, yes, part of the lymphatic system, but their role is specialized. It helps in the role of absorption of lipids. In the digestive system, we'll come back and talk about that. Okay. All right. So right now, you're just kind of focusing on the Peyer's patches, which is kind of integrated within the mucosa of the small intestine. All right, I'm going to go ahead and get a head start here and talk a little bit about immunity. I won't cover all of it, but that's fine. We got plenty of time. We'll keep working on it over the next couple of lectures. So when you think about immunity, there's 2 big divisions here. All right, 2 big classifications: innate versus adaptive. When you think about innate, okay, this is oversimplified, but we will use it. Innate is considered the non-specific defense mechanism, right? Okay, but we do know now, a lot of research does show it's not really as non-specific as we once believed it to be, okay? But overall, there's no receptors, right? So you're thinking about cells like macrophages and neutrophils that belong to the innate response system, where all it's doing is looking for something that does not belong in their body. Is it foreign? Is it non-self? And it's going to mount an immune response and take care of that target, right? As opposed to the adaptive response, which is mostly, we're talking about T cells and B cells here. This is very, very specific. Why is it specific? Because each T cell and B cell is programmed in a specific way. It has a receptor on its surface. It can only recognize one foreign pathogen, right? You got millions, billions, trillions of these B cells and T cells circulating throughout your body, correct? Each one of those B cells is programmed to recognize one specific antigen, right? One specific foreign antigen. Same way with your T cells. That's the biggest difference between the innate and the adaptive arms. Okay, innate, like I said, is very non-specific. So we're going to talk about different lines of defense, or like what are the components that make up the innate system, right? So anything, your first line of defense is not even like, it's just a boundary, it's a barrier, like your integument, right? That is your first line of defense, and it's part of the innate response. So just the fact that you have the epidermis there, which has like what, 50, 60 layers of remember the stratified squamous epithelium, that's your boundary that's preventing easy access of pathogens unless you have a cut or something like that, right? So that's your inner, that's part of the innate response, is it's your barrier, right? The mucosa of all of your internal organs, like the respiratory tract and so on and so forth, they're also part of that innate response, especially if they are modified to where they have cilia. You remember cilia? Hair-like structures? Yeah, so the trachea has ciliated epithelium on the innermost lining. So if there's any dust, pollen, pathogens, or anything that you have inhaled, it can kind of hold on to it, right, and walk it away from the lungs. So that's part of your innate response, right? We're not doing anything fancy. It's just a structural element that performs an immune role. You also have, in relation to the skin, I think this comes up a little later on, you have a lot of different secretions from the skin, right? So you got sweat, you got different antimicrobial kind of properties within sweat, like dermcidin, things like that. You have a lot of different antibodies. And then you also had sebum. Remember the sebaceous glands that release sebum? Okay, those oily secretions also have a lot of antimicrobial kind of properties. So all these secretions are great too, right? A lot of these secretions, especially sweat, it is at a lower pH. You remember that pH scale, 0 to 14, 7 being neutral. So anything 6.99 downwards is considered acidic, right? So pH of sweat is acidic. Acidic environments are not great for proliferation of bacteria. So that's your first line of defense. It didn't have to do anything fancy, just a lot of barriers already in place that's going to make it harder for infiltration of a foreign pathogen, right? Does that make sense, y'all? But of course, if you do have infiltration of a foreign pathogen, which we've all encountered, right? Then what do you do, right? I mean, then how do you take care of this target? So this is where we're going to say, all right, my second line of defense is this. Now I'm going to activate very specialized cells called phagocytic cells. These are your neutrophils and your macrophages, right? And then I'm going to activate other cells like your dendritic cells that are going to sound the alarm and activate more cells. And then, of course, we're going to talk about antimicrobial proteins, more in relation to like interferon proteins, like viral antivirals. Okay. Inflammation is actually a defense mechanism, which is part of that innate response. So that's a very important piece. I want to come back, and I don't know if I'll have time, but I'll definitely spend some time talking about that, probably next lecture, right? Then you have your adaptive response. So all of this is innate, non-specific, right? You got your barriers, your boundaries, and you got your activation of your phagocytic population of cells and so on and so forth. Inflammation. We'll also talk about something here called complement proteins as well. And then you have your adaptive response. Now, this is specific. Specific, you got your B cells, T cells. They're mounting a very specific response against one foreign pathogen, right? So typically, anything that's specific takes a little bit more time to prime, to actually activate. There's a lot of steps here, right? This is why when you first encounter a foreign pathogen, it's your innate response that jumps in first, right? I mean, because it's not specific, all it's saying is, I don't recognize you. I mean, this is a foreign pathogen. It doesn't belong in the body. I'm going to do a lot of different things to try to eliminate this while the adaptive response is getting primed and getting up to speed, right? So your innate response is non-specific, which is great in the sense it's faster to respond. Anything that responds fast, though, short-lived. Okay. But in the meantime, you are priming the adaptive response and it's getting there. So normally it takes, so when you first encounter foreign antigen, you feel miserable for, I don't know, a few days, 6 days, 7 days or so, right? Before you feel like, oh, now this is actually under control. It's turning around because it takes about 5, 6 days to prime that adaptive response to get those B cells and T cells activated. For the B cells to then get to start secreting antibodies. So your T cells to become all of these different populations of cells where they know what to do with that target, right? So your innate response is acting first. It's kind of giving you some time. It's just trying to do its best, right, to kind of contain the target. But at the same time, you have your adaptive response that's gaining strength to take over. Well, it's not really a takeover. They both work together. Okay. All right. So here's the, broadly speaking, here's your innate defenses, your surface. Oh, I did not do that. All right, hold on. It shut down my PowerPoint. Do that. All right, remote users. Uh, that's not it. Okay. Can y'all see? Yes. Okay, perfect. Thank you. Okay, let's do this again. That was, okay, I need to go all the way down here. All right, this is where I was. All right. So your innate responses are your surface barriers. You have your non-specific responding cells like your phagocytic cells, neutrophils, and macrophages. We'll come back and talk about natural killer cells. Inflammation is a very important aspect of your innate response. We'll also discuss antimicrobial proteins. You do spike a fever when you're fighting an infection. It's all related to the inflammation process. It is a good thing because elevated body temperatures basically are not very hospitable for proliferation or multiplication of those foreign pathogens, right? So it's a good thing to have. That's why you don't want to really have, well, if it is manageable, you do not want any fever reducing kind of medication because you really, that's a good thing for your body to fight that infection. Of course, you have to be wise about it too, right? I mean, like you're controlling the fever just enough, right? Your adaptive responses. This is where we'll spend a whole lot of time talking about humoral immunity, which is what the B cells are doing, and then cell-mediated or cellular immunity, which is what is mediated by the T cells. So I'm going to talk a little bit more about this today, and then the next two classes, we will break this down into humoral and cellular immunity, okay? So I discussed different surface barriers, like the skin, mucous membranes. Both of these play a role in providing a barrier and also all the different secretions from these two different barriers. Physical barrier, that's the integument. Remember how the epidermis has these specialized cells, the stratum corneum layer, the outermost layer of the epidermis, are very modified cells. They're all dead cells, right? Do you remember this? Where it's packed with a very tough protein called keratin. Right. So that's going to play a huge role in acting like a strong barrier. It's a physical barrier, it's a chemical barrier. It's also a microbial kind of a barrier, right? So it's a barrier that prevents easy access from the outside. Okay, then you have a lot of different secretions and chemicals related to those secretions. Like I said, pH of a lot of skin secretions, it's mostly acidic. This is called the acid mantle. This is going to be not so hospitable for growth or proliferation, multiplication of different pathogens like bacteria, right? You have different enzymes, like saliva has something called lysozyme. You've got, it's not here. Oh, it's right here. Dermcidin, that's part of sweat. All of these are different enzymes and different chemicals that play a role in, okay, it doesn't eliminate the target, but it slows it down. Sense, right? A lot of this is basically slowing it down, right? Till you get to actually your phagocytic cells where they can eliminate the target. You also have other modifications related to the mucosa of the respiratory system. Like, for example, you've got the cilia, like, okay, pointer, cilia, that helps to basically trap any kind of pathogens and kind of walk it away from critical organs like the lungs, right, to prevent spread of that infection. So these are your innate barriers. Then your second line of defense looks like this. And I'm gonna break this down on the board next class for you. But for right now, phagocytic cells. When you think about phagocytic cells, two things should come to mind immediately. Your first responders are neutrophils. Your second responders are those monocytes that differentiate into macrophages. Yeah, all these names should be familiar to you. These are all formed elements, right? So we talked about WBC population, where you have 5 subpopulations. You did a differential WBC count. You identified neutrophils are going to be your highest population of WBCs. You remember this? Yeah, maybe. Okay. 45 to 65% or so, right? Kind of like the neutrophils are 50 to 70% in that range. And then you have your lymphocyte population and so on. So lymphocytes are not your phagocytic cells. They are part of the adaptive response. Monocytes, 3 to 8% or so, right? Which then would differentiate into your macrophages. So I'm going to talk to you, remind you really, because we've already covered this in ANP1. I'm going to remind you how these two populations of cells work. Why are they phagocytic cells, right? They participate in a process called phagocytosis, right? Which is through vesicles. I'll talk about this. Then you've got natural killer cells and so on and so forth. Let's start here and see how far we can cover today. Phagocytic cells. Your first responders are neutrophils. When you're mounting an immune response, when you have an infection, when it's all part of the inflammation process, your first, okay. When you're fighting an infection, when you have a target to go after, right? You're going to hear this a lot. Remember with the clotting mechanism, right? There were some stress signals that were released, right? I mean, of course, you had the whole platelets that were inactive that needed to be activated. And then there's these chemicals that are released by the platelets, which allows more activated platelets. And then you have that plug being formed, right? So it's very similar to that. You first have to sound the alarm that we have recognized a foreign pathogen here that needs to be eliminated, right? This alarm is really part of the inflammation process. So there's a lot of different chemicals that are released that are saying, okay, we've got a problem, and that problem is here in this particular region. Everybody needs to congregate here, right? So this is where we're attracting cells, immune cells. We're talking WBCs here, right? So again, this is back in the red bone marrow. Do you all remember this? In the red bone marrow, you have a population of stem cells called hematopoietic stem cells. When you have all of these stress signals, when you have all of these red flags going up saying we got a problem here, right? What's happening? Back in the red bone marrow, we're receiving these signals, and that's prompting those stem cells to differentiate, right? Right now, I need neutrophils. Right? I need more monocytes that will then become macrophages. So what do you have? You have these stem cells that are committing to become more neutrophils. Is that right? Or committing to become more monocytes, or committing to become more B cells or T cells, lymphocytes. You see, all of that is happening. Okay, well, then they leave the red bone marrow, right? And then they start entering the blood and lymph, and now they home to where they need to be. Right? So now I got these neutrophils that are arriving at this site of injury or infection where I need to clean up that target, right? Okay, so what happens? Your neutrophils are your first responders. Within 4 to 6 hours, you're going to see a high number of neutrophils. Okay, so here's what's going to happen. A macrophage or a neutrophil kind of, okay, say it looks like that. It's going to kind of extend these processes, and these green rod-shaped structures, maybe they're bacteria, right? And so we are going to internalize that bacteria inside of this immune cell, this phagocytic cell, which is a macrophage or a neutrophil, right? This process is called phagocytosis. And how did this happen? The plasma membrane of this macrophage cell forms these vesicles. You remember this? It's an infolding of the plasma membrane. I'm going to grab all those green rod-shaped bacteria. I'm going to pull it into this macrophage. I'm going to pull it inside of this neutrophil, which is then where we would eliminate that target. And this is by binding with something, an organelle called a lysosome. Okay, so I'll talk about that in a second. About those macrophages, there are some macrophages that are fixed, meaning they don't move, right? I mean, they are resident macrophages. You're going to see this. Definitely come back and talk about this with the digestive system. Liver related macrophages called stellate macrophages. They play a huge role in screening the blood that comes through the liver, detoxification to prevent basically widespread dissemination of that pathogen. You also have a group of macrophages within the brain, and these are your defense cells within the brain called the microglia. These are supporting cells, right? So they are your policemen within the brain and the spinal cord. Then you have the majority of your macrophages that are not sitting in one place. They're constantly moving around. How are they moving around? They're moving around through the lymph. And of course, they're also circulating through the blood, right? So they're constantly moving around looking for a foreign pathogen. All right, so here's what's going to happen. We'll come back and talk about opsonization in a second. But this is the process of phagocytosis. So if this purple cell is a macrophage, or maybe it's a neutrophil, right? And I have encountered this foreign pathogen shown here in green. So what does phagocytosis look like? This, again, this is covered in cell transport processes, chapter 3. We covered this in ANP1. You may want to go back and review it, or this schematic ought to do it. So this macrophage or neutrophil is going to form vesicles. These vesicles are basically infoldings of the plasma membrane, which then allows you to internalize this bacteria, whatever it is, this pathogen, right? So then this vesicle is going to pinch off, becoming what I call a phagocytic vesicle. And what's inside this vesicle? Well, this is my target that I want to eliminate, right? So then this is going to merge with an organelle here called the lysosome. These purple dots inside the lysosome, those are very, very acidic, low pH, right? Low pH hydrolytic enzymes. When these enzymes, when this lysosome merges with the phagocytic vesicle, we're going to unleash those enzymes on my target. And what does that do? These enzymes are going, they're acid hydrolases, right? So they're going to chop up, they're going to degrade my target. This target happens to be a bacteria, for example, right? And then I'm going to spit out those contents safely. Does that make sense to you? That's how a macrophage works. That's how any phagocytic cell works. Process of phagocytosis. Is that right? What if this was a neutrophil? What if this was a neutrophil? All right, same process, right? We're going to internalize the target. The lysosome is going to combine with the target, and it's going to eliminate the target, except we call neutrophils noble warriors, right? They give up their life in the process of fighting that pathogen. So what's going to happen then is those acid hydrolases not only eliminate the target, they are also unleashed randomly, right, in the interior of this neutrophil cell. So they eliminate the target, but they're also eliminating themselves. Do you see it? Macrophages work the same way, except they don't eliminate themselves, all right? This is why neutrophils are short-lived. So they're the first responders. When you sound the alarm, they're the first responders. They're the cells that get released. They fight the target, and then they get eliminated. That's why their half-life is only 10 to 12 hours, right? So they do their job, and then you're going to start seeing it decline. But then macrophages come in right on the heels of those neutrophils, and they can kind of take over. I'm not saying the neutrophils will just die. I mean, they'll die off, but then you're producing more neutrophils, and the fight continues. I'm just saying it's short-lived, okay? All right, I will come back and talk about the rest of these elements of the innate response, and then move on to B cells, okay, in the next lecture. One more thing. I do not know what's going to happen related to the weather, so keep an eye on it. I don't know what CTC may or may not do. So if there's any campus closures or anything like that, not anticipating it, but you never know, look for an announcement from me. Okay, this is a high flex class, so if we do not have class on Thursday in person, I'll still meet you remotely. Does that make sense? So we won't skip a beat. Because I already lost about 15 minutes with you today. All right, we'll see you all. I'll see you all one way or the other on Thursday. Remote users, do you all have any questions for me? I'm so sorry about all of the delays and all of the technical difficulties we had, but we figured it out. So thank you for being patient. I'm going to, thank you. I'm going to let you all go. Have a wonderful rest of your day. Bye.